So yes, so my background is usually high resolution vessel structural imaging, and you might like, okay, the ICP, the P is not for structure. Why should we care, right? About vessel structure. I don't feel that much out of place because of the very first talk, which was excellent and already showing some of the variability in structure. But I want to go into the vessel structure more. Maybe to motivate why vessel structure. For example, if we look at the bicycle flow, maybe in a city, taking into account the infrastructure, you see there's a structure in the infrastructure. This might help to differentiate why maybe more people in Utrecht cycle compared to a city in Romania, right? I see somebody's taking a picture, so I wait until the next slide. Was a coincidence that I found this, and I think it's nice that it's not car related. So I think it's fair that maybe also in the cerebellum and the vasculature, there are certain maybe patterns which we really like to look at. Now, obviously, I'm not that smart, and other people have thought about it before, right? For example, Michael Garnier or Julia Hook, she was presenting on Friday. So if you have questions about these studies, you can ask her. But what they did is they built vessel atlases. So instead of just explicitly co-registering the structure with respect to each other, they also co-register the vessels explicitly with respect to each other to have kind of a normative distribution and a bit of variance and so on. Nice idea, very cool. There's another recent trend, I'm also from where I'm from, so I'm a bit exposed to that. But this is more from the kind of clinical side, where we say, well, there are sometimes distinct vessel configurations. Again, you could talk to Julia. She's presenting a poster where you can see that the circle of Willis has certain configurations. Maybe the PCOM is missing or something like that. But similarly, in the hippocampus, there are different vessel supply patterns. We call it like that. We can argue later on if that's really supply pattern or not. But the point is the PCA seems to be always contributing to the supply of the hippocampus, and in some, but not all subjects, there is the ACHA also supplying. And in case of a pathology, in this case, cerebral small vessel disease, patients who have like two vessels supplying cope better with cognitive decline. So you could call it a resilience mechanism, if you like. Again, we can argue later about resilience and reserve, but this is kind of the literature. Now, my motivation was, we have on one hand the vessel atlases, where you have kind of a unimodal distribution and everything is done automatically. And on the other hand, we have kind of this distinct patterns or classes which are done in manual fashion. And I want to basically convolve those ideas with each other, right? I would love to see, that's a hypothesis, could be wrong, if there are multimodal distributions of our vessels. And obviously, I don't want to do that by hand because I'm not an MD student, right? So I want to do something automatically. Now, at least I think there are three challenges which we need to address. The first one is the smaller the vessels, and we're going to look at the putamen, as the title suggested. The smaller the vessels, the more heterogeneous the vessel patterns are. And it becomes non-trivial to co-register all the vessels explicitly with respect to each other. You could imagine if somebody has 4 ventriculostriate arteries versus somebody who has maybe 3, it's hard to pinpoint which should be co-registered just with each other, okay? The other thing is how to move from these kind of discrete classes, which our trusty neurologist identify, to more continuum, right? You have before AB, and now you want to have something in between. Well, we see if we find something. And then again, the automatic part, because label intense work is not my thing. I have a solution, or at least I propose a solution. It's up to you to decide if it's really one, to combine structural normalization. So we skip the explicit vessel co-registration, and we learned that at least in the deep gray matter, we are good doing that. We can do we use an atlas, so lucky. Then we're going to use vessel distance mapping. I'm going to explain that, and non-negative matrix factorization. I'm going to explain that also very briefly in the next 2 slides. So these are saturated intensity projections of 5 hemispheres of the data we want to look at, of On top, the venous data, and on the bottom, the arterial data. And you see already that there are kind of similarities, but sometimes they're also quite heterogeneous. So first thing we're going to do is we're going to go to MLI space. Just, at least for me, it's visually a bit easier to compare. And you might see some common patterns. For the veins, there seem to be sometimes something like a at the anterior head, some vessel which might reappear in a lot of subjects. And if you look down here, it appears that the kind of amount of fanning from middle to the outside is a bit variable, but you know, each vessel sometimes looking like they're flipping you the bird, these angiograms, but at least in the middle is always something. Okay. Now, as a distance mapping, maybe sounds fancy, but the idea is super easy, and other people have used that. What you do is you segment the vasculature, and then for each voxel in your structure, you compute the closest distance to a segmented vessel. And it's a so-called distance transform. It's very easy. And we applied a number of metrics, but for example, this VDM metric we applied in the hippocampus. So there was an MD really segmenting all the vessels manually to make sure that we covered everything. We computed then the distance of each hippocampal voxel to its closest vessel. And we saw that in a mediation model, this works out quite okay with 0.72 arc squared. Showing again, we moved there from a kind of A or B classification to more continuum, because the distance on average for your hippocampus is rather continuous value. So this kind of fixes maybe the first two challenges. The last one would be the automatic pattern retrieval. And for that, we used NMF. This is similar to PCA, right? For PCA, you have your input matrix. And what you then do is you decompose your input matrix in smaller sub-matrices, which you potentially can better comprehend or maybe use for practicing, whatever you want to do. Now, as the name suggests, non-negative, compared to PCA, you have only positive elements there. The advantage, in my opinion, is there that you will reconstruct or you will represent your data as the weighted sum of certain patterns. And they only are additive in a positive sense, which makes the interpretation easier, because you never mix a pattern A and B where you maybe subtract something, and then a C where you add something again. It will only add on top of each other. And the other thing which makes interpretation a bit nicer is that we enforce, sorry, there's a spelling mistake, not enforce space components, or that sounds cool. It's sparse components with using orthonormal projections. And Robert et al., there are many studies out there using NMF, but Robert et al. Used it to look at microstructural variations in the human striatum. So they had T1 and T2 values and FA and D and so on. They constructed the input matrix, they decomposed it, and then they were able to get these kind of microstructural informed parcellation of the human striatum, which is completely unsupervised clustered. Okay, so maybe to the data at hand, we're going to use data which was acquired at the site IM, but before I was working there, the so-called study forest. It was more on the fMRI side, but just by accident, they had a 3 Tesla Presto sequence, which we then used for venous depiction, and a 7 Tesla TOF angiogram, which we used for arterial depiction. We co-registered everything to MNI and did a pretty vanilla vessel segmentation by using a Frangi filter and then an automatic hysteresis thresholding. So here we have the data, and we segment it. We compute the vessel distance map. So for each voxel, we know how close the next vessel is in millimeter. And what we do then is, per hemisphere, we have this kind of 3-dimensional map. We're going to flatten this into 1-dimensional vector, right? And we do that for all hemispheres to build up our input matrix. We concatenate them. Basically, each of these columns will be then one hemisphere. And then we use the magic of NMM to build 2 matrices out of it. The first one is the so-called component matrix, which is voxels times components. the number of components is up to me to decide, or to you if you want. And it will be, for example, if you have 1,000 voxels, it will be 1,000 voxels. Maybe if you pick 3 components, 1,000 by 3. What you can actually do then is you can select maybe the first column or second or third and reshape that, and you get the pattern in 3D back, right? That's very nice because you can then really visualize the components or patterns the algorithm found. And then we have the weight matrix, which is components times hemispheres. So for each hemisphere, it will tell you how it decomposed the data or how you can reconstruct it. For example, if hemisphere 12 has a rates of 0.5.5, you know, I have to take 0 of pattern 1, 0.5 of pattern 2, and 0.5 of pattern 3 to reconstruct my input. Good. And now let's look at some very, very preliminary results, just as a proof of concept. We see here the component matrix after reshaping it again. These are axial views through the putamen. And you can see, for example, this is pattern 3 for my arterial data. And I see the sparsity constraint actually worked. And this pattern seems to store the distances at the anterior head of putamen. So that means that if somebody has a very high weight for pattern 3, that means this will be scaled very much up. So that tells you there's a lot of large distances, so there's little vasculature there. So a kind of intermediate mental steps to do, but with a bit of brain gymnastics, you can make sense of it. Similarly, for pattern 2, it seems to represent more the posterior distances. And it's a bit of a different story for the venous side, where you have, for example, pattern 3 representing more superior drainage or distances towards that. And we can also look at the rating matrix. So again, on the x-axis, you have your hemispheres. We had 20 participants, so we have 40 hemispheres. And here the rate per component or pattern, whatever you want to call it. We see that it seems that most hemispheres have a rather same contribution from the pattern 1, but there's a distinct difference if pattern 2, how much pattern 2 is distributing to the input data. And one interpretation could be that even if you do some clustering, that you might have 2 distinct configurations of vessels, patterns, however you want to call it, whether they have a large posterior supply or not. Or no, it's not supply because we are not ASL, I have to be sensible. If the distances are high or we detected a lot of vessels here. For pattern 3, which was the anterior part, you see it's a bit of a mixed story. It's something in between. For venous data, there's already more variability in the very first component, which reflects actually what you also see if you look qualitatively at the data, that in some cases we simply did not detect a lot of venous vasculature, which is in part due to this fact that they used the Presto sequence to acquire it. I'm not sure if you're familiar with that sequence. It's basically, it enables you to acquire a TE, which is longer than your TR because it's a reverse GRE. Problem is you don't have flow compensation for that, which is not ideal for venous depiction, I would argue. Good. So I hope I could convince you as a proof of principle that we can do these kind of automatic pattern detection, and at least it seems to be feasible to marry the concepts of having a uniform distribution and maybe also distinct differences in the patterns, that we can be more on the gray zone between. Obviously, it's very preliminary. I need to do a quantitative evaluation. What number of components is really optimal? I picked 3 because 3 is a nice number, but that will not hold up in the review process, I believe. And also we need to interpret the patterns more with expert knowledge. As a simple-minded engineer, I might not be the best for that. But once this is done and everybody's more or less convinced, I'm happy to extend it to our brain region and then hopefully apply it in conjunction with vascular functional imaging to maybe explain variance in healthy physiology based on the structure which is underlying. And with that, I would like to thank you for your attention.